crack GAT-B and DBT exam? Biochemistry is the backbone. If you master this concept, you are halfway to your dream seats at a top institution. Let's dive in. Identify the amino acid that provides maximum flexibility to a polypeptide chain. Here is your countdown. The answer is glycerin because having only a hydrogen atom as its R group, it has least static hindrance. Next, which protein structure level is not destroyed during denaturation? Here is your countdown. The answer is primary structure because denature breaks hydrogen bond and hydrophobic interaction but leaves covalent peptide bond interaction. Next, which interaction is primarily derived of protein folding? Here is your countdown. The answer is hydrophobic effect because nonpolar side chain clusters in interior to minimize contact with water, increasing entropy. In Ramachandra plot, where are beta sheets typically found? Here is your countdown. The answer is upper left quadrant because this region represents the specific phi psi angles allowed for extended beta stance conformation. Which amino acid is a precursor for serotonin? Here is your countdown. The answer is tryptophan because tryptophan is hydroxylated and decarboxylated to form neurotransmitter serotonin. How does an uncompetitive inhibitor affect Vmax and Km? Here is your countdown. The answer is both Vmax and Km decreases because the inhibitor binds only to the enzyme substrate complex, shifting the equilibrium. Next, which enzyme classes catalyzes the joining of two molecules using ATP? Here is your countdown. The answer is ligases because ligases like DNA ligase forms carbon-carbon bond, carbon sulfate bond or carbon nitrogen bonds coupled with ATP hydrolysis. Next, what does a high Km value indicates? Here is your countdown. The answer is low affinity for substrate because it means a higher concentration of substrate is needed to reach half of Vmax. Which vitamin act as a precursor for FAD? Here is your countdown. The answer is riboflavin, vitamin B2, because FAD is a redox cofactor essential for TCA cycle and fatty acid oxidation. Induced fit model of enzyme was proposed by. Here is your countdown. Answer Daniel Koshland. Because it suggests the enzyme active site change shape slightly to fit substrate. I hope you enjoyed the video well. Drop your scores in comment section and do subscribe for more such videos. Let's evolve together.